Hello and welcome to part 2 of this video on microscopes and magnification. The previous video I went through standard form and converting from millimeters into micrometers and so on. And I also introduced the terms magnification and resolution. So if you haven't seen that video, please make sure you do so before watching this one. In this video I will talk through the differences between the light and electron microscopes and how to do calculations for magnification. So light microscopes work by shining light, you can see a bulb here, through a very thin specimen, and there are various lenses in this apparatus to help enlarge or magnify that specimen so that we can see it. So essentially you have to position your specimen on a platform which is called the stage, and this could be moved up and down, and the specimen is often placed on a microscope slide. You have your light source. In this picture, the light source is built in, so there is a bulb already in there. Some of you may be familiar with using a microscope where you have to position a desk lamp flat on the surface of the desk, and there'll be a mirror here that you can pivot to shine light upwards through the specimen, but both of which work in exactly the same manner. So light shines through the specimen here, and it has to go through one of these lenses here, and these lenses are called the objective lenses. And a lens is just a curved piece of glass that can make the specimen appear bigger, and they vary in their kind of magnifying strength. So they can vary between something like times 10 to about times 40 on a school light microscope. And then you have got right at the top another lens called the eyepiece lens, as this is the one that you look through immediately, and these are often set at times 10. So the total magnification of a light microscope, if I just write that down here, total magnification is the sum of the eyepiece lens and the objective lens. So if eyepiece lenses tend to be times 10, and the maximum objective lens you can get on a school light microscope is 40, the total magnification of a light microscope is times 400. So the image that you see could potentially be 400 times bigger than what it actually is. This is the coarse focus, and it basically moves the stage up and down. And this dial here is for the fine focus, the more precise focus. There we go. Right, so there are certain advantages and disadvantages of using this microscope. They are relatively cheap to buy, they are portable, and you can view living specimens. The specimens are live, and sometimes you can actually have the specimens swimming about and you'll see them nice and clearly. Some disadvantages then. So they can have a low magnification, namely they can go up to like times 400. Some industrial microscopes can actually go up to times 2000, but if you've got one of these at your school, you are very lucky. Along with having low magnification, they also have low resolution. So generally speaking, the quality is not particularly great. But as I said, they're cheap, portable, and you can see living things. Moving on to the electron microscope. These work by firing electrons at your specimen, and these electrons actually have very narrow wavelengths, much narrower than the wavelengths that you have with visible light. And as a result, you are able to see in a lot more detail. So they're more detailed. They can magnify something by approximately 2 million times its original size, which is pretty incredible, while maintaining the resolving power of just 0.2 nanometers. The specimen is set up so it's in a complete airtight vacuum. The temperatures need to be just right, the pressure needs to be just right, and as a result, those temperatures, those pressures, the fact that it's in a vacuum so there won't be any oxygen means that what you're looking at is a dead specimen, I'm afraid. You won't be able to see little amoeba cells moving around like you can with a light microscope. So they do allow us to see in a lot more detail. They are fantastic. but what you're seeing is dead, and unless you're a millionaire, you can't afford them. They are very expensive. They're huge, so they're kind of floor to ceiling machines, so definitely not portable. If any of you have the opportunity to visit a lab in a university, that'll be your only chance to see an electron microscope. You definitely won't see them at school. And actually there's two different types of electron microscope. There is a transmission electron microscope. They have the most detail, but you see the picture in 2D only. And then there are these really cool electron microscopes that are scanning electron microscopes. They don't actually allow you to see in as much detail as a transmission electron microscopes, but you do get a 3-dimensional image, and they look really, really funky. One of the required practicals is to prepare slides containing animal and plant cells to view under a microscope. Typically, the plant cells that are viewed are those of onions, and for animal cells, you simply do a cheek cell swab. To create the plant cell slides, you need to use tweezers to peel off a single layer of onion skin, so this will be the epidermis of the plant. And this epidermis, which is very thin and transparent, is laid flat on a clean microscope slide. In the case of the animal cell, a cotton bud is used to swab the inside of your cheek to extract the cells there, and the cotton bud is then rubbed onto the centre of a clean microscope slide to transfer the cheek cells to it. With both plant and animal cells, the cells are transparent, which creates a problem when you're trying to view them under a microscope. To overcome this, a stain is used to pick out the features of the animal and plant cells. In the case of the onion, iodine is used as a stain, and in the case of animal cells, methylene blue stain is used instead. In both cases, a mounted needle is used to slowly lower a cover slip over the stain. The cover slip keeps the specimen flat and retains the liquid underneath it, but it must be lowered slowly to avoid any air bubbles getting trapped underneath it. Now the slide of either the plant or animal cell is ready to view, and it is viewed under a light microscope, initially under low power. This is so that you have the widest field of view to find the cells that you then want to zoom in on at a high magnification to see in more detail. Once you've got the cell in more detail, you then need to draw it and label some of the features of those cells, and it is worth including the total magnification used to view those cells under. So if you're looking at the plant cells from the onion skin, you may see something and draw something that looks like this. Here the nucleus, the cell wall and the cytoplasm are labelled. With the animal cell, from the cheek cell swab, you may see something that looks like this. Here the cell membrane, the cytoplasm and the nucleus are labelled. The cell membrane isn't actually visible under a light microscope in either of the plant or animal specimens. However, the cytoplasm and cell membrane do take up the methylene blue stain differently, so you can distinguish a barrier between the cytoplasm and the liquid mount behind the cells. However, you cannot actually distinguish between the cell wall and the cell membrane in the plant cells. So even though a cell membrane would be present in the onion skin slide, it is better to actually label the cell wall because that is detectable with a light microscope. So in this part of the video, I will be talking you through how to calculate the magnification of a cell under a microscope. To do this, you need to appreciate this formula, the I-AM formula triangle. So the I stands for the image size, and the image size is referring to the photograph or the drawing size that you might see on your exam paper. The A stands for the actual size, and the actual size is often a small measurement in micrometers if we're looking through, let's say, a light microscope. And the M stands for the magnification. So when you are asked to calculate one of these factors, you should be able to access the other two bits of data. So let's look at this particular example. You have a bacterial cell, measures 1 micrometer in length. I'm just going to cross this out a second, and let's say it measures like 6 micrometers in length. Calculate the magnification of this image. So we are looking for the magnification of this image. We are given the actual, but we are not given the image size. So for To do this, you need one piece of equipment, which I'm hoping you will always bring into your exams, a ruler. And you have to position your ruler so that it is lined up alongside the image of your cell, and you are literally going to measure it at its longest point. So we're going to measure this in millimeters. That's 10, 20, 30, 40, 50, 60, 70, 80, 90, 111, 115, 16, 17, 118 millimeters. So for our image size, we have got 118 millimeters, and we have to divide that. This line here, by the way, means divide by the actual size, which we have been given as 6 micrometers, but 118 millimeters is not in micrometers. We need to convert it into micrometers. You have to multiply it by 1,000. So when you do the maths, you get this: 19667 times. So this drawing on this screen is 19,667 times bigger than the actual size of that bacterial cell. In the next calculation, you are asked to calculate the actual size of the nerve cell's nucleus. And you have been given the magnification. So you're trying to find out the actual size. You've been given the magnification. You need to use your ruler once again to work out the image size, but not the overall image, just the image size of the nucleus. So we bring in our ruler and we line it up again to measure the diameter of the nucleus, which is, I would say, 9 millimeters. So that's the image size equals 9 millimeters. Remember, we have to convert that into micrometers, so we'll just add a few zeros there. So we times it by 1,000 to turn millimeters into micrometers. So we have got the image size over the magnification, which is times 500. And you do the maths and you get 18 micrometers. So the diameter of this nucleus is 18 micrometers. In this example, you need to use this scale bar to calculate the magnification factor of this image. So cue our ruler again. This time you want to line your ruler up with your scale bar. So we have got, and it's in micrometers, 10, 25, 26, 27, 28 millimeters. So 28 millimeters, times it by 1,000 to turn it into micrometers. Divide that by the 6 micrometers that scale bar equates to. Do the maths and you get 4,667. So this cell has been magnified 4,667 times. So moving on to example 3b, use your previous answer, which is 4,667 times magnification, to calculate the actual length of this sperm cell. So here we're calculating actual length again. And it's the full length of this sperm cell, so the longest stretch really. So let's get that ruler again. Let's line it up. Okay, so it's in line there, and it's in line there. So we're talking 183 millimeters in length. So we've got 183 millimeters. Times that by 1,000 to turn it into micrometers. So we have got our image size now, and we have to divide that by our magnification, which we found out on the previous question. 4,667 and we get the answer 39. It's actually 39.2, but we'll just round it down to 39 micrometers. So you might actually get a question that goes straight to asking you this: calculate the actual length of this sperm cell, and they'll give you a scale bar, and you might find you get a bit stuck, but remember that scale bar is there for a reason, and you could measure that scale bar using your ruler, divide the image length by the actual length to work out the magnification factor, and then you're on the home run really, because you've got everything you need to calculate the actual length of that sperm cell. Okay, well, good luck on those questions. They are a bit tricky. I hope you've enjoyed this video. Please check in again.